I'd like to welcome our audience to what is going to be a fantastic panel discussion on the latest prevention and treatments for COVID-19 that are in clinical trials. More Health, where I am Chief Medical Officer. This will be the fourth panel discussion we've had on COVID-19, but the very first on prevention and treatment. These are purely educational. There's no commercial attachment for this seminar, and I am just so pleased that we could get the panelist and moderator that we have. I'll let Dr./Professor Sarah Doernerg, our moderator, introduce herself, and then she will introduce Drs. Tebas and Osinusi. So thank you all for attending.
Great.
Dr. Doernerg.
Hi, everyone. Thanks for having me. I'm going to start off, I'm going to just show a brief presentation, and then I'll introduce Drs. Tebas and Osinusi, who are going to give the bulk of the talk. I can just introduce myself briefly. I'm an infectious disease physician here at UCSF. My day job is doing transplant infectious diseases and clinical research on antibacterial resistance, as well as running our antibiotic stewardship program. But as with many people, it's all COVID all the time currently. And we're just going to give a brief overview of COVID-19 before I turn over the floor. So let me get here. So this is a roadmap of what we're going to talk about today. I'm going to give a very brief summary. I'll introduce the speakers. Dr. Osinusi will give a brief talk, and then Dr. Tebas, and then we're going to leave a lot of time for discussions and questions. So I think many of you are aware of these numbers, but there have been at this point, more than 5 million COVID-19 cases worldwide, with over 300,000 deaths. Each time you update the information each day, the numbers continue to grow. It's been really astonishing. In the United States, these are the new cases as of yesterday, and you can see we continue to have a lot of new cases every day, despite maybe some flattening of the curve that we're seeing. And I think the topic of today is really how do you approach a new disease with all of a sudden a lot of patients in the setting of a pandemic, with patients in front of you who need treatment now, but without a lot of information. And I think there's a lot of things that are floating around, antivirals, anticoagulants, convalescent plasma, repurposed drugs, immunomodulators, and then new approaches. And how do you sort through all of these things in real time and figure out what the best thing is to do for patients? And that's been, I think, one of the biggest challenges. I was going to just mention kind of what's happened here, and I was discussing with Dr. Tebas before this that in the Bay Area, we've been quite lucky to, perhaps by a combination of luck and a very proactive Department of Public Health, to avoid a big surge. So perhaps we've had a bit more luxury to think through our approach to trials here. But we have a trials committee that at our 3 hospitals reviews trials and considers which ones we're going to bring on. We take into account things like the scientific rationale, the existing background data, other trials that are running concomitantly so we're not overlapping too much, and then opportunities for addressing different populations, so mild disease, serious disease, different phenotypes, as well as feasibility. And then one of the things that I think is becoming more challenging, perhaps as the pandemic slows down slightly, is which patients go into which trials and how to kind of choose the best path for each patient, and that's been a challenge here. Some other challenges that we found is certainly enrolling patients with an infectious disease in which PPE is of limited supply, is trying to figure out how to enroll patients without having contact with them, without visitors being there in the setting of patients who are in the ICU. There are new workflows, new floors of the hospital that are open, so nurses that may not be as familiar with the study groups. Many studies enrolling with different stakeholders. And then it's hard to do quote nothing. So a lot of people are wanting to give experimental therapies without being in studies, and that's complicated some of the enrollments as well. I think some of the good things about doing studies during the pandemic is we are generating data very quickly. And that's been a really exciting story. Many people are really engaged, so it's been an opportunity to bring together a lot of teams. And I think for patients, it gives them some agency in a time when there's very little control over what's happening to be able to participate in a trial. So I'm really looking forward to the presentations today, and I'm going to start to introduce Dr. Osinusi, and we'll let her share her slides while I'm introducing her. So Dr. Osinusi is the Executive Director of Clinical Research. She leads the Emerging and Respiratory Viruses Group in the Virology Therapeutic area at Gilead. She has over 15 years of experience in infectious diseases, liver diseases, public health, and drug development. She served as a project or clinical lead for multiple products that have made a massive impact in the field, mostly in the field of hepatitis, Sovaldi, Harvoni, Epclusa, that have really helped to revolutionize this field. And her current role is the project team lead for Remdesivir, leading efforts across various indications, including COVID-19. Anu trained as an infectious disease physician at the University of Maryland, after which he led the hepatitis branch clinical research and subspecialty medical services for the DC Partnership for HIV/AIDS Progress prior to joining Gilead in 2014. She has authored over 60 scientific publications and has a passion for rapidly advancing transformative therapies. I look forward to hearing her talk.
So, I think, just as, Sarah nicely alluded to earlier, I mean, I think it's important when we talk about the story of Remdesivir to actually think back to where we were just a few months ago. And I think, the involvement and how we got into COVID-19 was really, I recall the email sent December 31st by one of our, virology experts who works very closely with the WHO about these few mystery cases of pneumonia that were happening in China. And, based on some of the work then with Remdesivir that had been done for, other coronaviruses like SARS and MERS, he had reached out just to, give us a heads up, like, these cases are coming through. And then, of course, there were the case reports coming through from the WHO. And so for us, it was, okay, it's this new coronavirus, and we know that Remdesivir has activity against SARS and MERS in vitro, and we've already had some work with Remdesivir in other disease areas. Like, let's think through, what could be coming down the pipeline. And then, the novel coronavirus was identified a few weeks later by the Chinese CDC. Towards the third week of January, we had the first US case, and then just moving forward, the WHO declared this as a public health emergency. But it's important to note that at that point, January 31st in the US, there are actually only 6 cases in the US. Fast forward through March, a lot of activity, which I'll cover, but then, this was officially declared a pandemic. At that point, there are now more than 100,000 global cases, and in the US, there were 1,200 cases with 40 deaths. Fast forward a month later, worldwide, 1 million infections. Towards the end of April, it's now, over 1 million infections in the US, and where we are really today globally, these numbers are from a few days ago, with more than 4 million infections, more than close to 300,000 individuals have unfortunately died. And in the US, we're close to the verge of hitting 100,000 deaths. And so, it's just moved so rapidly in such a short period of time, and thinking through how do you develop a therapeutic for a disease that no one knew about until end of December? The clinical profile wasn't really clearly known at the onset, but trying to think through how do you answer the questions that are going to be most important and most impactful for the larger population going forward. And so where we were really when we started our development program for Remdesivir in January, where we had two key important information data sets that allowed us to really accelerate the program really quickly. And first was the non-clinical piece. So Remdesivir itself, it's a broad spectrum antiviral agent. It's a nucleoside analog that inhibits the RNA-dependent RNA polymerase gene in many different virus families. And what we knew about the coronavirus family was that the initial report showed that there was a lot of sequence homology in the polymerase gene with SARS and SARS-2, which causes COVID-19. And so what we knew was that we had a lot of in vitro data and some in vivo data from animal models, primarily rats and non-human primates with both SARS and MERS. So we had that data set in there that could suggest that maybe this would work for this virus. And then on the clinical end of things, before you can just go into into any disease population, and which is some of the challenges with this pandemic around new investigational therapies, there are multiple steps you have to take to get into patients that have a disease that is ongoing, and a key part of that is safety. But for Remdesivir, because in its prior to this, we had been developing it for actually for Ebola, and had done multiple studies in pandemics with going on with Ebola. And so at that point in time, we also had a safety database of over 500 individuals, largely individuals who had acute Ebola virus disease, as well as healthy volunteers, that showed that the drug was safe and tolerable, which was able to help us accelerate into a new disease area very quickly. Okay. And so, our three strategic priorities really were: the goal is to determine safety and efficacy and ensure that we have fast, broad access for patients. And so, on the clinical end of things, just collect all the clinical data that is important to be able to get this, have a good, the information needed for safety and efficacy to know if a drug works, and then be able to accelerate the path to approval. In addition, manufacturing, which I'm not going to really talk about today, for Remdesivir is extremely complicated. It's more than 70 different reactions, and it's a long lead time. But quickly for us, it was important that while we didn't know if the drug would be effective, with a lead time that is 12 months, you have to plan way in advance, hoping that things would be successful, but you can't wait till you get the readouts to actually start escalating your manufacturing process. And then also ensure access. And I think a big part of access is also looking at the populations you actually treat in your clinical trials, and make sure that you have a diverse population of patients, in which I'll speak to later, to ensure that if the drug does read out as effective, at least you're then able to ensure that it's applicable to a large population of patients. And so for us, the key questions we wanted to really answer is Remdesivir safe and effective for COVID-19? And if it's effective, what duration of treatment is really needed? Recall, we're going into a new disease. When you look at respiratory virus therapeutics, most things in that space have been all designed around flu therapeutics. And when you think about flu, it's typically a 5-day treatment. For coronaviruses, how long do you have viral shedding? For this new disease, how long is viral shedding? If you shut it down, can you actually treat for shorter durations? But when we were going in, all we had was data from our Ebola trials. And our Ebola trials were 10 to 14 days of treatment. So we wanted to really know, did you need that long treatment, or could you actually shorten treatment? And so a lot of our initial trials have been around addressing these two questions. And so I think, some of the other key considerations that you need to think about is which populations do you study first? As we've seen with this pandemic, you know, the stepwise fashion of, you know, it's people with very, very early disease. But if it's too early and most people recover, then do you really need a therapeutic? If it's extremely sick people with an antiviral, we know the earlier you treat, the better. And there are other mechanisms that have kicked in with people with ARDS or being on a ventilator, and there are multiple other things going on beyond the virus, as we've seen with flu and other respiratory viruses. And does it work at that stage? And recall at this point, we didn't really know much about this disease. Now we know that unlike influenza or some of the other respiratory viruses, they actually shed much longer. And so people are shedding for many, many weeks, even when they're on a ventilator. So maybe an antiviral might still have a role. But I think those are still all questions that we need to tease out. And then I think the other key piece was which endpoints are actually clinically meaningful. When you think of Tamiflu, for instance, you know, you look at the trials, and yeah, you do reduce symptomatology, but you look at the viral load endpoints, and you didn't really necessarily see drops in viral load in oseltamivir versus placebo, but you had a clinical benefit. And then mortality has never been an endpoint used in respiratory therapeutics. However, we know that with this, you know, with this new disease, you know, a big burden in the healthcare system has been patients who get very, very sick and go on ventilators and then unfortunately pass away. But what is clinically meaningful? Is it that you come off a ventilator? Could be very meaningful. Is it the duration of hospitalization? Is it duration of fever or some of these other clinical endpoints? I think that was also one key part to consider. And then, of course, I think, you know, with any antiviral you're developing, you think about viral load testing. And I think as Sarah alluded to earlier, when it comes to viral load testing, I think part of what we've learned about this disease is essentially that, you know, a lot of the tests up front, when you're thinking of nasopharynx or oropharyngeal, is all sampling upper respiratory tract. And we know upper respiratory tract viral load peaks in the first week and then goes down, but then you have a lot of lower respiratory tract disease. And someone having pneumonia, how useful is a surrogate endpoint of nasopharynx when it's not really clearly demonstrating what's going on in the lung? So I think those are some of a lot of things that we thought about as we're designing these trials. And then, of course, you know, part of the complications that happened, and it happened in the US and other countries as well, was all around the accessibility of viral load testing when, you know, for sequential monitoring versus when you have limited supplies and people are actually using it for diagnostics. And then, of course, other considerations, heterogeneity in clinical practice and management across board. As we've done all these different studies, you see countries where steroids are used in 2/3 of the populations, very different practices in the US, different practices in parts of Asia. And then, you know, logistic challenges in global shutdown when there's restriction of non-essential personnel that usually help with research studies, and having to do a lot of these activities remotely, and site personnel being pulled into clinical duties and also still doing research, and you know, shortage of testing supplies. So I think these are all things that we considered as we were developing the programs. And so just moving on really quickly into that key aspect of which populations should we really study first. Again, as I said, is it a mild to moderate? Is it people who have severe disease? Is it the critically ill? Should we have cutoffs of how long people have been sick, especially in a disease we don't know much about, and say, like, you know, if you've been sick for more than 1 week, an antiviral wouldn't work, but we don't really know because this is a new disease. And then I think a critical piece that we ran into very early on was compassionate use programs, where typically that happens in the setting of you demonstrating proof of concepts that something works. And then you could have, you know, a compassionate use program before you actually even get approval. But in this case, the burden of cases and the requests that were coming through were in a scenario where we actually hadn't demonstrated proof of concept yet, but in the absence of no other therapy and the hypothetical or the knowledge that we knew from the non-clinical program, we were faced with this very early on about having a compassionate use program. And so this just really outlined some of those efforts very early on. I think, once that first US case was diagnosed, not too long after that, January 25, we got our first request for compassionate use based on the non-clinical data that we had then published, and the fact that there was no other therapeutic out there. So we actually dosed our first compassionate use patient. And just for people who are not aware of what that actually means, so these are individual single patient use protocols where a physician has to make a request for a patient and then, go through all the clinical information for that one patient, sign a whole bunch of forms and, consent the patient, have a protocol in the US, and different regulatory patterns exist in different countries. But for instance, in the US, you call up the FDA, get an emergency IND authorization that allows you to treat this patient with an approved drug. And then on our end, we go through a lot of processing and paperwork, and then you are actually going to ship that individual drug to that site, get it on a plane or wherever it's going, and ship it to that hospital, and then someone picks it up there and transports it to the hospital. So you have to go through this very manually intensive process for each patient that you do this. And so that's, that was a compassionate use program. And then not too long after that, we initiated our studies in China. And I think it's important to note that when these studies started February 6th, 99.9% of the cases of COVID-19 were in China. And so, typically, most times in most companies, your phase 3 program is an internal program. But in this case, we felt that this was the right thing to do. 99.9% of cases were in China. And so we started this investigator-sponsored phase 3 studies in China, looking at two populations, mild disease and severe disease. And then not too long after that, we started the NIAID study. And it's important to note that in collaboration with NIAID, and it's important to note that these 3 studies were all double-blind, placebo-controlled randomized trials. So that could really get to that question of safety and efficacy. And then in March, we started the Gilead severe and moderate studies. And these studies were really designed to look at that duration aspects of 5 days versus 10 days. And they were called simple studies because, again, there was this need to move very quickly. And many times when you're designing like large phase 3 studies that you're trying to set up central labs and do a lot of these typical things that you do, the startup process takes 12 to 14 weeks. But in this case, from concept to execution of those studies was 3 weeks, because there are a lot of things we shaved off, like requiring local labs or just running things much faster in a pragmatic way, recognizing these are hospitalized patients, healthcare workers are stretched very thin, and there are just some critical aspects of what you need to get done that you need to actually include in these trials. And then in addition, we've initiated studies with multi-global organizations like INSERM and then the WHO. I'll talk a little bit about the compassionate use PEDS pregnancy piece next. And then a lot of these studies have started reading out and have now been published, like the China studies were published in April in Lancet, and hopefully in the next week or two, in the next week or the next few days, the study results from some of these other studies will be published as well. So just again, highlighting that piece I talked about with the multiple mechanisms that you put in place to support pre-approval. Again, as I said, our first request was January 25 for the compassionate use, 1 patient. And then we started getting a whole lot of cases, not only in the US, but in Europe and in Japan and globally. And again, as I said, for each individual patient, you go through this process and ship the drug to each individual provider for each individual patient, and not a mechanism really built to handle the volume of requests that were coming. By March 21, when we transitioned the program, we were getting more than 300 requests on a daily basis. for compassionate use. And with that volume, no matter how many people you throw at it, because it's a manually intensive process, then there are delays. And these were patients already hospitalized, very, very sick. I felt like that was not the best mechanism to deal with that issue. So we quickly transitioned to an expanded access model where at least you have the drug on site at an institution. So when a patient comes in and needs to be treated, you can offer the patient that mechanism. What we actually then did with the individual single compassionate use program was we felt that it was important to continue the program. And the program has continued in pregnant women and pediatric patients. Because while there is a recognition that the disease burden in pediatrics is less than adults, however, we also know that as we get new information from this program, we know that extrapulmonary manifestations like Kawasaki disease and a lot of other things have been shown in pediatric patients. And we felt it was important to not wait till you're done with all your trials or your development program to start actually including pediatric patients. And the same thing with pregnant women, because pregnant women were also getting the disease, and we were getting requests, and we felt it was the right thing to do to actually continue this program in pregnant women and patients under 18 years of age. Okay, so moving real quickly to the endpoint question. I think there are many key considerations, again, like, we know that patients treated early in disease with an antiviral will have a different course. And what's that ideal timing for outcome evaluation for a disease that is new? Is it 7 days? Is it 14 days? Is it 21 days? Is it the same for people who have pneumonia alone, or is it you look at a different time point for people who are on a ventilator? Because each time points that you look at and the power you build around looking for efficacy is going to be very different, and so you have to be very thoughtful as you select some of those time points. And then we also felt it was important to look at clinically meaningful outcomes that capture patient function, recovery, or death, and look at outcomes that are very efficient, evaluations for efficacy. And I just put this there not to say that everyone should look at these different scales, but I wanted to put this out there because this is just an idea of just the different scales that are being used in multiple different trials. It's really the ordinal scale, a lot of it built around work from influenza. And as you can see, there are different scales. So like the 6-point scale was what was utilized in the studies in China. The 8-point scale is what is in the NIAID study. The 7-point scale is what's in the Gilead study. And now there's a lot of overlap about these categories, but you can see that there are differences. Also, when you look at the ordinal scale by itself, it's actually more helpful than looking at some of the other endpoints, but it also has its own caveats in the sense that, is a 2-point drop from someone who's on invasive mechanical ventilation to come off intubation, come off high flow oxygen, and then just be on low flow, the same as someone who starts on essentially no oxygen and is discharged. Like, is that the same clinical benefit? It's hard to say that it is, but it's at least it's a thoughtful way of thinking through some of the key questions to consider. And with that, I just put this up there just to show some of the endpoints that are there. Like even with this ordinal scale, you see that time to clinical improvement by the 7-point ordinal scale, or recovery, which was what was is now being utilized in the NIAID study. Our studies look at distribution on that scale. Some studies are looking at inpatient hospital mortality. And I really just put that out there just to highlight the challenges that are, with the different endpoints. But I think, what most investigators have now done is recognize that as we've gotten more information, there's just some endpoints that make a little bit more sense than others. So I think early on, there was endpoints that were just looking at fever and normalization of oxygen saturation. But I think after a while, like, 2 weeks in, as to the publications came out of China, what you recognize is while 90% of people have fever as a presenting symptom, when you actually get into the hospital, it's only 47% of people have fever at hospital presentation. So using fever like as an endpoint in itself might not necessarily be very helpful. But I think, in general, there's just been a consensus to try and approach not only looking at, the 2-point null scale, 2-point improvement that is going to be reported in a lot of studies, but also looking at things like recovery, which means you actually come off all oxygen support or you're discharged, or looking at duration of intubation or duration of hospitalization or duration of oxygen support. So while there might be one primary endpoint, a lot of studies actually have additional exploratory endpoints that they're looking at as well. And then I think I'd like to just highlight this one slide as well, which is around the viral load testing with antivirals. I think, what we've learned is that results vary based on the assay you use, the collection sites, the quality, the timing of illness and mutations. And it's very important, again, to try and understand even more as we go ahead, what's the optimal duration to sampling, taking out some of the viral sampling challenges. How do you correlate viral shedding with disease severity, especially now we have case reports of people who get better, they're discharged, and then you can pick up shedding somewhere else. And then with a lot of tests that we do, which is viral RNA, what are you actively measuring? Is it active virus? Is it fragments of virus? And then how do you really correlate, like in the hospitalized patients who has real pneumonia, short of you doing bronchial alveolar lavages or getting really good induced sputum, nasopharyngeal samples in a disease where we know it goes down after that first week in the upper respiratory tract and it's all now lower, how do you do that, and what value do you place on viral shedding versus a clinical symptom or a clinical outcome? And then, of course, the risk to healthcare workers as you do repeated testing, and then the shortage in testing supplies I alluded to earlier. So I think this is probably my last slide. And again, just to highlight, there are a lot of other studies ongoing right now with, some studies are ongoing right now with Remdesivir, but I just wanted to highlight the key ones. And I think, the first two, again, as I alluded to earlier, were the studies in China. The good thing was that this study started very early. However, which is also a great thing, is that the epidemic was quickly controlled in China. And over time, what happened is that those studies did not enroll sufficient numbers of patients. So the first study, which is a severe study, ended up enrolling just over half of the patients it was designed to enroll. And so that reduced the powers to detect a difference significantly. So the study became an underpowered study it was only 58% powered to show efficacy. And so the data available from that study was inconclusive. The second study has been suspended because it's only enrolled about 25% of patients it was supposed to enroll for similar reasons. And then the next study was the NIAID study. And I think what you can also see is what populations each of those studies were targeting. So the NIAID actually did target all three populations of moderate, which is essentially that you have pneumonia, you're in the hospital, but you don't have hypoxia. The severe group, which is you're hypoxic, and then the critically ill group where you're intubated. That study read out with top-line efficacy demonstrated, and it was a placebo-controlled study and will be published online by NEJM, the New England Journal of Medicine, later on today. And then the Gilead simple trials have also, one of the studies has read out, which is a severe study. And what that has shown is that the 5-day treatment course is just as effective as the 10-day treatment course. The data from the moderate study, which has a standard of care arm, will be available over the next few weeks as well. In addition, there are large studies which Sarah alluded to earlier as well, the thing about COVID-19 is it's really fostered a lot of collaboration across multiple countries, regions, and the huge WHO studies and interim studies are going to enroll, over 10,000 patients. Our studies, both the severe and the middle moderate studies are going to expand into the Part B, where they're going to enroll thousands of patients, with the severe study over 5,000 more patients, the moderate study close to another 1,000 patients as well, which gives us a lot of robust data to be able to come up and look at additional subgroups and look at the efficacy in more detail. And so I think actually this is my last slide. I just wanted to say again, when you fast forward to the pink part, the compassionate use program, which started first, was published in NEJM mid-April. The NIAID positive results, phase 3 Gilead severe study were released, press releases end of April, and will be published shortly. And at this point in time, we're moving forward with regulatory approvals. In the US, we have something called the Emergency Use Authorization granted, which allows more easier access for a drug before it's for use in an emergency setting like a pandemic, but it's not an approval. And then in Japan, we actually do have full approval for Remdesivir as of May 7. And we continue to pursue regulatory approvals across the globe while ensuring that we're increasing manufacturing and continuing to get this readouts from all these different studies. And so I think that's my last slide, and I will stop now. And the back to Sarah.
Thank you so much, Dr. Osinusi. We will come back to you with questions. I think we're going to hear from Dr. Tebas first, and then we'll come back to you at the end. That was a wonderful presentation. All right, Dr. Tebas, while you're pulling up your slides, I'll introduce you. Dr. Tebas is a professor of medicine at the Hospital of the University of Pennsylvania. His research interests outside of COVID center around the study of the complications of HIV and treatment of HIV. And currently, he's very interested in strategies to cure HIV infection. He is the director and the PI of the AIDS Clinical Trial Unit at the University of Pennsylvania, and is one of the founding members of the HIV Reservoirs and Viral Eradication or Cure Transformative Science Group of the AIDS Clinical Trial Group. He's also involved as PI of several early phase trials of new compounds developed in the conjunction with NIH related to HIV. And I really look forward to his talk on prevention of COVID-19 today.
So I'm going to talk to you today, and thank you for the kind introduction. And I'm really honored to be in this webinar. I'm going to talk about a project that I'm working on mainly, which is developing a DNA vaccine against COVID-19 in collaboration with the Wistar Institute, the University of Pennsylvania, and Inovio Pharmaceuticals. And Dr. Osinusi already talked about how quickly things have evolved over the last 5 months of this epidemic. You have to remember at the beginning of this, and this is a wonderful slide from Dr. Wu from the Chinese CDC, what happened really happened in a matter of 4 or 5 weeks. Basically, the first cases of COVID-19 were reported at the end of December. In Wuhan, immediately there was an alert, epidemiological alert. The Wuhan market was closed on January 1st, 2020. The WHO was informed. The virus, the new virus, the SARS-CoV, was identified 1 week later. Immediately, the WHO, the sequence of the virus were shared with the WHO, and they were published only a few weeks later. And the first PCR tests were developed. And at the end of January, the city was shut down. I'm going to change to a laser pointer so I don't get my slides too crowded. And the epidemic has spread since then logarithmically. There has been, this is the screenshot from the Hopkins dashboard, and there has been more than 5 million cases in the world. You look at the US, we have more than 1.5 million cases of this infection, and it has spread all over the world. More than 300,000 people have died, and we are going to close very soon the 100,000 deaths at the US. And it has had devastating consequences for the world and the economies of the world and the people, all humankind, basically. This is a slide about the clinical manifestations, also from Dr. Wu. I really like this slide because it shows the main clinical symptoms of the disease, and either it's presented in mild, severe, or critical cases with fever, dry cough, fatigue, and less frequent symptoms. And also that the presentation in this part of the slide can be mild, moderate, severe, and critical. And in general, you have to remember that most people do well, most people do recover, and most people that present with mild disease have a complete recovery or a moderate disease. You can see also in this slide, as a small fraction of people progress, and they can be progressed to death. And that's the type of patients that I see in the floors or in the ICU when I work in ICU, taking care of these patients and testing therapeutic interventions to see if we can change the outcomes. We have learned a lot over this very short period of time about the SARS-CoV-2 virus, which is the cause of the COVID-19 disease. The virus has always represented like this. This is graph from the CDC, has spikes, which is red in the CDC graph. That is the protein that the virus uses to bind the receptor in the cell, which is the AC2 receptor. And that the virus is endositosed and creates a vesicle that acidifies and then releases the RNA. The way that hydroxychloroquine theoretically works, nobody really knows if it works or not, probably not very well, is by blocking the acidification of that vesicle. The virus releases the RNA and it starts translated into proteins and replicate in the RNA, and then almost in a reverse process matures through the Golgi and the reticulum, and then is released from the cell, and the cycle of the virus is completed. The body fight the viral infection, creating an immune response. So the virus is ingested by these antigen-presented cells, and a peptide is presented to those cells, to the T cell helper. This is a very simplified schema of the immune response, and then the immune system mounts a B cell response with secretion of antibodies, some of them binding to the virus, some of them neutralizing the virus, and we will talk about those two concepts later. And then also it creates a cellular immune response that people think both the combination of these two immunological responses are critical to, one, control the virus and provide long-term memory, both with antibodies and T cells, to prevent new infections of the virus, although we still don't know how long-term that immunity built after natural infection is, how long that that resistance will last. Still much to learn. Many different groups, there are very different technologies that are being used to develop vaccines against the coronavirus, and the classic approaches to vaccine development is to use live attenuated virus or kill virus or proteins from the virus that are injected, and then the immune system responds to that injection, mounting an immune humoral or cellular response to that injection. But of course, those type of approaches to develop vaccines take a long time, because you have to grow the virus, isolate the virus, separate the proteins, purify the proteins, making sure that there is no contamination there, that if you're using an attenuated virus, it's completely attenuated, and that you cannot infect people by giving the vaccine. The technology that has become very popular and powerful, although there is no vaccines approved using nucleic acid vaccine approach. And basically, when you try to do this, is when you know the genetics of the virus. When you have information about the virus, you can put that information of the protein or the part of the virus that you want to test and develop immunity against. You put it in a vector of DNA, and you create a plasmid, a circle of DNA that has the gene of interest. And for coronavirus vaccine, the gene of interest is usually the spike protein of the virus. And then you manufacture these in bacterial cells. You purify the DNA, and you inject in under the skin or intramuscularly. And in order for the cell to uptake this plasmid and transfer it to the nucleus and start making proteins, that is the ones that are expressed in the dendritic cells, on the antigen presenting cells, and that generates the immunity. In order to increase the efficiency, is almost sort of an adjuvant for the process. You give an electroporation. It's an electrical current. It's a small electrical current that increases the uptake of the cell. And the way I think about it is it creates small holes in the wall of the cell, and it increases the uptake. And it's the same idea of as an adjuvant. You can also have nucleic acid vaccines using mRNA, and this is a very popular approach now. This is the approach of Moderna Pharmaceuticals. Probably many of you have heard about this. And then you do basically the same thing. You put in a piece of RNA your gene of interest, and then you inject that into the individual. It goes into the cell, and you start translating proteins. And the same idea. You have expression of the protein of the antigen of interest, and your body generates an immune response. You don't give electroporation with these vaccines, but you have to coat the mRNA, you have to coat it with a lipid coat, and that might complicate a little bit the manufacturing of these, because it's lipoaffecting. It has special formulations, and it has to be, it's a little bit more complex manufacturing, but allows also for a rapid manufacturing, probably being a little bit faster than a DNA vaccine. As I said, there is no approved vaccines in humans for any of these two technologies. There are a couple of vaccine approved, DNA vaccines approved for the use in animals, or for DNA vaccines. The approach of the vaccines that we are working now at the University of Pennsylvania is a DNA vaccine. And we did that in collaboration with Inovio and the Weiner Lab that is at the Worcester Institute. We have worked together before to develop vaccines against other emerging pathogens like MERS-CoV and vaccine. That's a paper that has been recently published in, I think, in Lancet of Infectious Diseases. And then we also work with this technology to develop these GLS 5700 in collaboration with Inovio, and also with GeneOne for a Zika vaccine that we published in the New England Journal a couple of years ago. And based on this technology, CEPI awarded Inovio a grant to support the development of a vaccine against the coronavirus, the COVID-19, in January of 2020. And that's what has been the main funder of this project. So this is a vaccine. It's called Inovio Inno 4800. It's a synthetic DNA vaccine that targets the spike of the glycoprotein. Literally, this is the data for preclinical development. This paper was published yesterday in Nature Communications, and talks about the development of the vaccine. The vaccine antigen that we selected to put in the backbone of the DNA is the spike protein of the virus, which is this thing that basically, these spikes is what gives the name to this class, this family of virus, coronavirus, because when you look under electron microscopy, it looks like a crown of a king. And these spikes are made of this protein. These proteins are the target of neutralizing antibodies, which are considered the main goal. This is what you want to develop in people to protect from subsequent infection. There is a technology that basically optimizes the codons and the mRNA for expression and for not other cross-reactivity And this part of the protein in yellow is what is called the RBD, or the receptor binding domain. This is the part that the virus uses to bind the AC2 receptor that I was talking about before. So the AC2 receptor binds to the SARS-CoV spike in this area, and this is the target of the neutralizing antibodies. When you vaccinate using the whole spike, which is what we're doing, you develop antibodies that bind to the protein, but they don't necessarily bind to the RB2, to the receptor binding domain. And this was developed basically in Dr. David Weiner's lab, and a bunch of people working the development of this vaccine based on the previous experience with the Zika vaccine and also with the MERS-CoV and HPV vaccine. This technology has been. So this is the idea of what is done when the virus is isolated, and the Chinese group published the sequence of the virus. You can basically clone the part of you pick the protein or your antigen of interest, and in this case was the spike, and most of the vaccines that are being developed using this antigen, and you clone the backbone vector you clone the spike of the virus in this backbone vector. We clone two different, three standard, and then an outlier of this virus. There was an outlier of these spike proteins. And then you have to do experiments in vitro. The first experiment is that when you put them in cells, you have expression. This is a DNA vaccine. You have expression of mRNA. And in this figure, what you see is the ratio between the acting mRNA and the mRNA of RNA of the driven by this plasmid. And you can see that there is expression of both plasmids when it's transferred into the cell. That's the first step. Then you have to prove that when you do that, the cells in tissue culture, they express the protein. And this is a Western blot just showing the protein band with the spike protein. And then you do immunofluorescence just to see in the tissue culture that the protein is expressed, and also it goes to the surface of the cell. And these are cells that have been transfected with these two plasmids, and then you stain with an antibody, immunofluorescent antibody, to show that those cells, and this in green here, are expressing the protein of interest. And that's what we hope the immune system will develop a response against too. Then you do in vivo studies in animal models, and then you give a single dose of these the plasmid with the spike protein, and you use Selectra. So Selectra is the battery, the device that gives the electroporation. So you inject the mice continuously, you give the electroporation, and you show that the mice develop antibodies. And this is just given two different doses of the vaccine, 2.5 micrograms or 10 micrograms. And the use electroporation, this is the titer that after 2 weeks, the mice develop the titer of antibodies, 1 to 10,000. So you still can detect the antibody when you dilute the serum of the mice for 10,000 times. And this is the vector without the insert, and you see that there is no expression, no antibody development. And here in this part of the figure, you're looking at interferon gamma T cell responses. So it's just to show that when you do this with DNA, you not only generate this kind of immunological response, the humoral immune response, but when you do DNA vaccines, you generate also a cellular response. And as you can see, this is the interferon, this is a ELISpots, and you are counting the number of T cells that are reactive to the proteins of the virus, the epitopes of the virus. And then you can count the colonies, and you can see different pools of epitopes because the interferon gamma release with different peptides, and you see that there is a strong cellular immune response that is generated in the mice. You also do the same thing in other animal models. The guinea pig animal model also is reactive. Is a good model to test the antibody production with this vaccine. And as you can see here, you can see high titers of antibodies produced in this guinea pig model. So when you give the vaccine in vivo models, the animal models, you see a robust cellular and humoral immune response measured after a single dose. For this antibody, you use an ELISA plate. You put the spike, and you see there is the presence of antibodies that bind the spike of the virus. But that's not the only thing. Binding antibodies are important, but other antibodies, the neutralizing antibodies, are the ones that are the most important. People think that that's the right endpoint in clinical trials testing vaccines. So neutralizing antibodies are antibodies that bind to that receptor that the virus uses to enter the cell and prevent the infection of the cell. And you test it in monolayers of cell, you add the serum from the animal, and you see if you can prevent infection. You use also a pseudovirus assay because you cannot use live virus because it requires a BSL-3 or a BSL-4 lab. But you can do with a pseudovirus. It's a virus that expresses the protein, but the spike, but it doesn't. Is not the SARS-CoV-2 virus. And as you can see, and you are looking at the inhibition of the infection, and you can see the mice that were vaccinated in blue here, they have less infection over time than the mice that didn't receive the vaccine. So the vaccine in animal models generates neutralizing antibodies. And then what you want to do also is you want to see if those antibodies that have been generated prevent binding to the ACE2 receptor. This is the receptor that the virus uses to enter the cell. And if you have an immune response that is neutralizing, you will prevent with those antibodies the binding of the ACE2 receptor. And this is a curve of you use a peptide and you look at the curve of binding to the receptor, ACE2 receptor, and then you see if you can block that binding with the immunoglobulin, the antibodies generated after the vaccine. And you see if you have not given the vaccine, you don't have any blocking of the binding to the ACE2 receptor. And then if you use the serum from animals that have been immunized, you have this blocking. This is another way to look at this development of neutralizing antibodies. And it's nice that you can see this type of response after a vaccination. So the antibodies block the binding to the ACE2 receptor, which is a good thing. And then when you have all the animal data and there is an ongoing non-human primate models to try to see what happens when you use this vaccine and if you can prevent infection in the non-human primate model, but the data is not ready yet. Although, for example, for the MERS vaccine, it was clear that a similar vaccine prevented infections with the MERS virus, which is another virus of the coronavirus family. Then you start thinking about clinical development. And the first study that you do when you want to develop a vaccine is a phase 1 study. And what you want to look is at safety and immunogenicity. Is this vaccine safe? If anything happens to the people that get vaccinated? And do you generate immune responses in those people? And you do that in healthy volunteers. And normally it takes time to do this. It takes sometimes months up to a year to optimize the vaccine and to optimize the manufacturing and to optimize the antigen. But because we are all in a very rapid process with this infection, this is what it took to develop a vaccine that went to humans. So the sequence of the virus was published at the end of January of 2020. So all of these studies that you have seen were done between February and mid-March. At the same time that the studies were being done, the product was being manufactured and it was released for GMP. And an IND was submitted to the FDA on April 6th. The FDA approved and said that it's okay to proceed with the vaccine. And the same day, I mean, before we had written the protocol, submitted to the IRB, and getting it approved and put it in the clinicaltrials.gov, the same day that the FDA approved, we started to enroll participants in trial. First, you enroll one participant, and then when you have a DSMB meeting, you can enroll the second participant. And basically, we complete the enrollment of the 40 participants that we wanted to enroll in 4 weeks. And the enrollment of the phase 1 study was completed at the end of April 28th. And the last week 6 visit, which is 2 weeks after the second dose of the vaccine, it will happen at mid-June, and we will have some data about the immunogenicity and safety of the vaccine, hopefully at the end of June of this year. This is an incredible amount. With the Zika vaccine, it took us 7 months to get into the clinic. Now you're talking about 2 months. For Moderna, it was almost a week faster than this timeline, because the mRNA technology manufacturing is a little bit faster than DNA. And this is a study design. It's a very simple study, 2 groups, 20 people. The dose is at week 0 and at week 4. You give 1 injection or 2 injections at each visit, and you give a total of 2 or 4 milligrams of DNA. The injection is a subcutaneous injection. You inject it in the deltoid area, and then you use a machine to put these needles and give a small electrical current, and that is the electroporation. And this is an old experiment that shows that when the keratinocytes and the Langerhans cells, after getting an injection of a DNA vaccine that express GLP, and you can see that there is an uptake expression of the GLP after electroporation in different tissues of the skin, fibroblasts, keratinocytes, and Langerhans cells, adipocytes, and myocytes. There are multiple cells that get the DNA and express protein after the electroporation. So this is the schedule of events in this trial, and this is where we are right now. We are vaccinating the last, I think, 5 or 6 people next week, early next week, on Tuesday, I think. Everybody will be here. So this is where we are right now. The primary endpoint is at week 8. 4 weeks from now, we will have the primary endpoint, and we will be able to start analyzing the immunological responses of the vaccine. And then we will follow people for up to a year to make sure that the immune response is safe. And this is a collaboration of multiple groups. Inovio is the pharmaceutical sponsor. The Worcester Institute is the home of Dr. David Weiner. The clinical trials unit is at the University of Pennsylvania. CEPI was the funder, and the Bill and Melinda Gates through CEPI funded this project. And I want to say a special thanks to Jo Quinn and Sue Kim, who helped me to basically complete the study, and they were in contact with the patients, and they have been incredibly busy, but they have been incredibly effective in completing this study. And that's all I wanted to say. Thank you so much.
Thank you, Pablo.
Thanks so much. All right, I think we're going to kick off questions and answers. Marc, do you have anything you want to say first?
Yes, I am very grateful for Pablo and Anu for agreeing to give their presentations. And frankly, I don't think we would have been able to get other discussants that could do a better job, because you are conducting the trials, you know in intimate detail the agents that are being tested, and you have a significant background in conducting these clinical trials. So as CMO of More Health and speaking for More Health, we are very grateful. And I won't start off with my questions. So we'll let our moderator, Sarah,
all right.
that.
I've got a bunch of questions to ask you, and I focused less on the scientific nitty-gritty, which I think, as Dr. Osinusi says, you can access relatively easily, but more kind of on your opinion of how things are unfolding and challenges and good things that are coming out of this. And for the participants, feel free to type into the Q&A, and I can read your questions out as we go. So first one is just a general question for the two of you, which is, how did you get involved in COVID-19 research? I feel like we all had kind of like our pre-COVID lives and our COVID lives, and I wanted to hear how yours became a COVID life.
Anu?
Okay, sure. I'll take this one first. No, I think, for me, my background again is it's in infectious diseases. At Gilead, I oversee our emerging and respiratory virus group, and this was the emerging and respiratory virus virus that came upon us all. And I think it was just it's been 24/7 involvement, and not just only, I think for something of this size, I think it's so impactful, not only in our professional lives, but our personal lives. And it's a global pandemic that I think that even within the team here at Gilead, or when I talk to my colleagues in academia, like everyone is involved in COVID-19 research in some way or the other. It might not necessarily be a therapeutic or a vaccine, but looking at other organ systems that are involved, or infection control or supply management. Like, there's so many things about this pandemic that is making us rethink everything we do, not only in the research end of things, but how we live our lives and how we even practice as clinicians or do other things. So I think everyone is involved. I feel like it's really been that way for most people.
I think my experience in this hospital was that we, at the beginning in January, February, we all thought, well, this is happening in China. We'll see what happens. Although people were talking about how likely that this was going to spread quickly, and the Chinese were telling us that too, based on the previous SARS experience. It felt far away at the beginning. And then you were going through these preparations in the hospital, and you were thinking, well, maybe, are we overdoing this? And then when they start coming, when the people start coming to the hospital, and they were really incredibly sick. Some of the first people that came into the hospital, there were people that you have worked with, and some of them were younger than I am. And you felt very vulnerable, and you felt the need to do something about it. And I mean, I have done clinical research all my life. My love have been doing things with about HIV, and I have been doing that for all my academic life. And also, I always like emerging infections and trying to solve these emerging problems quickly. And I had a long-standing collaboration with David and to help develop clinically some of the things that they do in the lab. So it was a natural fit. And then I know the people in Innovium, we started working together, and we developed the trial, and we do clinical trials for HIV. This is not a big jump. But then I continued to be involved in clinical care of patients, and I was an investigator in the NIH trial with the Remdesivir, and we were seeing people in the ICU, enrolling patients all the time. And we were a site also of the 2 Gilead trials. We have enrolled, I think, around 80 people in those trials. And it has been a very enriching experience and very humbling experience, and definitely has changed our life, the way we live, and the amount we sleep.
Well, thank you both for your contributions. It's really inspiring. I'm going to switch the order of these questions. So I want to hear from each of you, what is the best thing about doing research during the pandemic? And then I'm going to follow that up with what is the biggest challenge?
So there are many things that have been the best, both clinical and in research. So clinical, for me, has been to discover how engaged people are in the hospital. To me, I have discovered the people in the ICUs, the nurses, They really went in to take care of patients. Of course, people were worried about their safety and worried about their families, but it was an amazing experience to see people not even thinking twice. I lived through the beginning of the HIV epidemic, and I remember how much stigma and rejection were to patients with HIV. People not wanted to go into the room, and I never saw that in the ICU. There were some people outside the ICU that might have some reticence at the beginning, but that faded away too. So it was amazing to see how the hospital and the nurses and the people that clean, the people that bring the food to the patients have changed. And that makes your life very easy. At the beginning, I went into the patient room to collect swabs because in the NIH study, that was one of the virological endpoints. After doing that 2 or 3 times, the nurse would come and say, "You don't have to do it, and I will do it because I have to take care of the patient." And that was incredible. And then the whole university, which can be a very slow machine sometimes in reviewing protocols or getting things done, focus completely on getting these trials moving. And the IRB was calling and approving submissions and reviewing submissions in a matter of 2 or 3 days. And so the whole institution became very focused on making life easy. That was the best thing I see for clinical research. And that has been my experience.
Yeah, mine too.
It's incredible.
Anu, do you have anything to add?
No, I think while I'm not in the hospital anymore, but I think it's really just in many similar ways. It's just the amazing shared collaboration that you see not only internally, here at Gilead, but across, working with the study team for the Acts 1 study that Pablo is a part of. Like, that was just an amazing collaboration, and just the speed of everyone working towards this common goal of trying to identify vaccines or therapeutics that work for patients for this pandemic was incredible. And I feel like, hopefully some of those collaborations that are being fostered or lessons learned, we can take this with us, and it doesn't just end when this is over. But we come up with new, creative, inventive ways to do things and move things really quickly for the common, larger good. I think for me, that's also been, the biggest part of this all.
Yeah, and I think has never happened in the history of medicine. This thing of going from a drug that has not an approval and develop the clinical protocol and completing the clinical protocol in 2 months. And getting an answer in literally 2 months. You're talking about more than 1,000 patient study, randomized, blinded, done in multiple sites, multiple countries, multiple continents, and getting an answer in 2 months. And then the Gilead trials is 5,000 people, more than 5,000 people enrolled in the same amount of time in all over the world, with the common goal of finding a way to treat people. To me, has been incredible, and I don't think it has happened. And this country and the whole world should be proud of being able to do that. Ask a question, answer the question, and then move to the next question. I think that has been something very, very unique about this epidemic.
Yeah, and I think just to buttress that point, I think the only reason where we are today, whether it's with vaccines or some of these other things, it's really because of this close partnership that has to exist, with government, academia, other organizations, these are things that, as Dr. Tebas said, takes years. The fact that we've been able to do this in months, it's just the importance of building those really strong scientific relationships with both the government, non-governmental organizations, and it's been invaluable, I think, in this pandemic.
How do you think we can keep ourselves from going back to our silos when this is over and going back to the status quo of the IRB taking weeks and things taking years to enroll? How can we keep the momentum going?
I think you need to qualify, because I don't think we're really sure if and when this will be over.
Yeah, I think the challenge is still there. I think the next huge challenge, on top of developing new approaches to treat the infection, is going to be the vaccine. And this effort of 5,000 people in a study or 1,000 people in a randomized trial that is blinded is really nothing compared to what we are going to have to do for a vaccine. 35,000 patients. I mean, it's going to be thousands and thousands of people that have to participate in trials.
So I'm curious to hear what the biggest challenge is. Is that the biggest challenge? Do you have others that you see?
Well, we tend to think that a phase 3 trial is going to be the big challenge, which is not the big challenge. It is a big challenge, but it's doable. And this country has done it before. I like history of medicine, but if you remember, in the 1950s, the polio vaccine was developed by Jonas Salk, the largest study ever done by humankind. 1 million kids were vaccinated, and there was a control group of 1 million kids. As difficult as it is, that is not the challenge, because I think we will be able to do a vaccine trial, and we will prove that one or several of the vaccines in development works. But then we have to manufacture billions of doses of the vaccine. Billions. We have to vaccinate 6 billion people to make herd immunity and prevent it and make this infection die out. And that's not a medical problem. That becomes an engineering problem. There is not the manufacturing capacity right now in the world to manufacture as many vaccines as we're going to need. And I hope there are good engineers out there that are thinking about how are we going to do this? How are we going to create billions of doses of a vaccine in a safe way that people feel confident that they can get the vaccine and they feel safe about it in a record time, which has to be a matter of months. And that's a problem. That's the big challenge. The next big challenge.
Anu, what do you think?
Yeah, I mean, that's I think that is definitely a huge challenge, because, you know, as Dr. Tebas said, like it's not only, I mean, it's one thing to even have effective treatments or an effective vaccine, then you have to always consider the manufacturing aspect of things, which, you know, with us similar as well. Like, you know, we have this limited drug supply, but we're working towards, you know, working very quickly to expand manufacturing, and we've been doing that since January, essentially. But I think, you know, some of the other things beyond that is, even when you have an effective vaccine, how do you ensure it's deployed, and, you know, misinformation that is out there, and, you know, there are a lot of other challenges to the systems. And I think, you know, the aspect of this pandemic that I feel like it's brought up is that there needs to be a new way of doing things, not only how we do research, but how we do things in manufacturing as well. Like, how do we leverage things across companies or partnerships that ensure that fallow space in manufacturing plants are being used for something, or there's synergies to the system. And then I think, of course, with the public health system and how we approach infectious diseases or our public health system, I think this epidemic has laid bare in many countries globally that we need to do things in a different way. But when I think about the smaller challenges, I think, you know, just in the thick of things, a constant challenge, which was also, you know, exciting, because it's like you're learning new things that on a daily basis. The sheer volume of the number of clinical trials that are ongoing for COVID-19, or you think back to January with the first few case reports, and then the first few series of 20 patients, and then the 100 patient report, and now boom, boom, it's like every week, hundreds, literally hundreds of papers, and the information changes so much, like, and you're trying to react, but not be reactive. So you have to be forward thinking about things that you actually don't really know much about, or see where it's going to go. And so that was a constant challenge, but also made you think of ways to be creative and innovative as you think through some of those things. Because I think, you know, everyone always has these questions: why didn't you do this, or why did you pick this endpoint, or if we knew everything we knew now, we would have done all these different studies a different million ways, but you just kind of go with what you know, the information you have and try and think through it. So it's been a challenge, but I think those are like some small challenges compared to, as Dr. Tebas said, some of what would be ahead for us in the vaccine space.
And also in the therapeutic space. I mean, how do you deliver vaccines to therapeutics to 1 million people? I mean, manufacturing at that speed, a complicated manufacturing process like Remdesivir is not. I don't envy your company's job.
Yeah.
So I have a question for Pablo, and that is, what is going to be the endpoint or endpoints for approval for vaccines?
Vaccines are not approved on immunogenicity data. They are approved on prevention of disease.
Right.
And when you have a situation that there is no many incident cases, then your trials have to be very large. The problem when you develop vaccines for emerging infections is that you get to that point that you're going to do the phase 3 trials, and by that time the epidemic has disappeared. That happened with Zika. It has happened to some degree with Ebola because you use these very effective public health measures like masking and washing your hands, and sometimes the epidemic goes away, and that makes the perspective of developing a vaccine for pharmaceutical companies not economically not very sound, because when they say, okay, we're going to invest all this money and then we're going to get there and then disease will have disappeared and we don't have a vaccine. We don't have a product that it can be used, and that's challenging. And, you were saying what is the endpoint? The endpoint in clinical trials or approval of a vaccine is efficacy, and efficacy means preventing infection and preventing disease or death or admissions to the hospital, and that's a challenge. When the epidemic, which is a great thing, that the epidemic dies off, it becomes very challenging. We don't know what's going to happen with this coronavirus, and maybe we have a second wave or a third wave, and then might be easier to prove that the vaccine prevents clinical disease, but we don't know what's going to happen, and that's the challenge.
I'm curious to hear, Dr. Osinusi mentioned a little bit the pace of information that we're all getting about this infection, and I've noticed that there's been this real strong movement towards rapid dissemination of material in preprints and news media, and sometimes that's caused problems, at least locally here, where someone prints out a preprint and says, we have to be doing this, you know, this drug showed promise in 10 patients, and all of a sudden everyone wants to do something. I'm curious about your thoughts on the upsides and the downsides of data being so readily disseminated, oftentimes before peer review.
Yeah, no, I mean, I think it's a brand new world out there, and it's been challenging in its own ways. However, I do think that, you know, it's really just about, I almost feel like even agnostic of medicine, it's the way the world is going. Like, you know, everyone wants to know everything immediately. And there needs to be, you know, maybe a more democratic process of ensuring that data is accessed on a global scale. I think the challenge when it comes to things like medicine is that we, at the forefront, like we're scientists, and things need to be thought of in a scientific manner, and the body of evidence and, you know, the burden of proof, which sometimes I think, you know, while some news organizations are not really talking about like the standard traditional journals now, you know, some of the blogs or the websites are trying to make sure that, you know, information is out there, but there are a lot of caveats that sometimes I feel doesn't exist, And then that gets into the general public, and then there's this expectation that is maybe not true or could actually in some cases be harmful downstream, that I think it just needs to be some degree of responsible journalism. But I do really appreciate the push by a lot of journals right now to ensure that, you know, articles are free for, you know, like any article around COVID-19, which ensures that people across the globe could get information very readily. And I think a lot of journals are reviewing things very, very quickly, and trying to ensure that at least, you know, peer-reviewed articles are published very early and it's free. But then a lot of people get a lot of information from non-scientific journal sources, and I think, you know, people just need to be a little bit more responsible about how some of that information is presented, and not always like it's breaking news. Like it's not like the breaking news thing for medicine needs to be a little bit different from like breaking news about like a political story. Just try and get the facts right, and, you know, ask the investigator or ask the people involved. So you release it. It might be a few hours later, but I feel like sometimes you see this urge around like breaking news, like it was politics, but this is medicine and it's people's lives, so we always need to be responsible.
I think it's a double sword thing. We used to think about journals and the peer review process as the way, okay, it has gone through peer review and that means that the quality of the science is better or is good. Now, everything that gets pre-released, you have to become the critical person. You have to become the reviewer and say, is this scientifically sound? Is this powered enough? Is this has the adequate controls? The kind of questions that we do in journal clubs, now you have to read, every time you read something, you have to ask the same question, but before you trusted a little bit of the review process, and then that has become your review process. And I mean, I like everybody, MDRX, we all look at those publications, and they are very important. And now people are doing press releases, and you're going on press releases, and soon we're going to go on tweets. Somebody's going to say, this is my work, and it's a very slow. We need to, I think you need to step back and take a good look at what is presented and decide if it is scientifically valid. And you basically, you have to review every single thing that you read and ask yourself, is this well done, and should I change my practice based on this? And be very critical. I think we need to learn to be critical and learn to reject things that the fact that it has been put in print, that doesn't mean that it's a good scientific paper.
There's another side to that. That is Elsevier, the publisher Elsevier. They have a large number of journals, medical journals. You cannot get the articles unless you, whether you're academic or not, you cannot get their articles online unless you pay for it. You have to, the institution has to have a subscription, and individual users cannot access the article. So not only is there too liberal publication, but there's a flip side. And for a disease this important, to have the policy of your publication, no one can read it unless they or their institution pays for it. It's not good.
I think that's going to be a side effect or a good thing that's going to happen with this pandemic. The public, the open publications is going to become the standard.
Yeah.
It's going to be very difficult. It's going to be very difficult for a journal to go back to where the information was not public. So the business model for journals is going to have to change.
Yeah, agreed.
I'm curious to hear your thoughts on how we're doing trials. You both mentioned that there's been a lot of good collaboration and coming together of various groups. I'm wondering if we should be even expanding more. Dr. Tebas, you mentioned we're going to have to immunize a huge number of patients to be able to see efficacy of a vaccine. should we be enrolling patients in settings other than kind of the traditional academic medical centers? Should we expand the way we do clinical trials more than we already have? And do you have a vision for how that would look?
I think I am a control freak, so I like phase 1 studies because I can control everything. But for phase 3 studies and big studies for these vaccines, we're going to have to think outside the box. And I think our references should be, as I said before, historical references. How the polio vaccine was developed. It was basically a nation effort. So when Jonas Salk had the polio vaccine and did this massive trial, or Sadin following year did a massive trial in Russia with a live attenuated vaccine, they were vaccinating kids all over the place, in schools, in gyms, all over the place. And we are going to have to think outside the box in this very highly regulated environment. How do we do that? How do we vaccinate 300,000 people? Because the clinical trials infrastructure is just not there. I mean, there is the engineering problem of manufacturing millions of doses of vaccines or pharmaceuticals. And then there is the clinical infrastructure problem. How do we do that? And this, I think the clinical trials will have to be very simple. And is you get vaccinated, and then there is a way to register you. If you have COVID-19, you basically, you're collecting endpoints that way. And we are going to have to think in a very different way of how we do efficacy trials. And it's going to have to be very simple, but massive trials. That's how I think it's going to have to happen.
And I think, I agree. I mean, I think that even for us, predating, COVID-19, like, with our HIV franchise, like our work in HIV or hepatitis C more recently, I think a lot of our studies we would always have the mix of academic centers, but a lot of like community centers as well, because a lot of patients go to those centers as well. And, sometimes the speed of getting things done in academic centers sometimes can be very challenging in some of these highly competitive development programs. But then I think, when you then think of like, COVID-19 as well, I think we faced a similar scenario in the sense that, our first studies started like in the US, where these compassionate use cases, and the compassionate use cases were popping up in community hospitals in California, community hospitals in Seattle, hospitals in New York. They're not like, it happens everywhere. And so by the time we started the Gilead simple trials, we recognized that, look, this population of investigators are going to be a little bit different from maybe the investigators on some of the other trials. And they're not going to be as versed as doing clinical research. And then you do have this scenario where people are stretched extremely thin. So if we ask that you enter every single adverse event and concomitant medication in someone in the ICU, and you end up with like 300 pages of records, like there has to be a fine line. And also recognizing that as you're training people remotely, you can't do your traditional investigator meetings or your SIVs. And so these are not maybe as experienced investigators that as you're typically used to. And so you have to have a lot of those conversations with the regulatory agencies, because I think on one hand, maybe there is a push to like, let's simplify things to get the answers done. But then there are a lot of regulations around developing products, right? Because we always have to worry about safety and make sure we're not cutting corners, because while we want to move fast, we don't want to make mistakes that happened in the past. So there has to be like a two-way dialogue, because if by regulatory law, you have to report 1 million things, like every single thing, and you can't really get that done in a pandemic setting where people are stretched thin, then you have to really figure out like what is the you need to do, and how can you be creative and thoughtful in those settings, and recognize maybe you have to do some additional things along the way over time. But I think it's almost like a two-way street.
Yeah, you have to be incredibly careful, particularly with vaccines, because you have a public that is very sensitized.
Yeah.
So when Jonas Salk did the study with the polio, and it proved that it was effective, the government licensed the vaccine to 6 companies. I mean, if you read history, and one of them, it was the Cutter company. It was a manufacturer in the Rockies. They made a mistake on killing the polio virus, and they injected kids with polio the following year. And there were cases of polio the following year. And that basically probably was the beginning of the anti-vaccine movement, when people think, well, the vaccines are not safe, you know, we don't know what you're getting our kids. That cannot happen with the coronavirus vaccine, because then we will have another wave of epidemic. So we cannot cut corners, but we have to make the studies simple. We have to make them avoid, okay, I want a swab from the right side of the nose, the left side of the nose, and now I want, it has to be simple. But we cannot cut corners. I agree with Anu. It has to be rigorous, and it has to be done safely, because if we don't do it right, and there is a safety event that for one reason the vaccine, something bad happens, we will lose the trust of the people, and that's bad. But the worst thing is that a lot of people will die because we didn't do it right, and that's not the right thing to do. Not because of the vaccine, it's because people will not get vaccinated, because we will create an environment that people mistrust the vaccine.
Yes.
Marc, how are we doing for time? I think you're on mute.
15 minutes.
Okay. All right. I'm curious to hear from each of you. What are the 3 most influential findings since the beginning of the pandemic?
Pablo, do you want to take that first?
Well, I will say that so far the most influential finding is that there is a therapy that works. And I really think that it was very tough to take care of these patients, and it's still very difficult to take care of these patients. And I felt a little bit hopeless for a long time. I mean, the standard of care is supportive care, and it works. And we learn a lot of how to take care of these patients. We learn that you can ventilate prone, and then the oxygenation improves, that if you use anticoagulants, you decrease some of the complications maybe. We have learned many things along the way. If you intubate early, people are going to do better. We learn that people, sometimes they have to be in the ventilator for weeks at a time. But you feel a little bit like you could not do anything. And I think when you have a therapy that works, that changes completely. It's like HIV, you know? At the beginning of the HIV epidemic, we love to take care of the patients with HIV because that's why we are physicians. But when you start having antiretroviral therapy, and then when you really have potent antiretroviral therapy, that changes completely the way you deal with things. So I think the development of a therapeutic intervention has been, or to me, one of the big things of this, having something to offer to the patients, and that you know, because the trial was done, that it makes a difference. I think that has been, for me, the most important development so far in this epidemic.
Anu, what do you think?
You know, I would say I agree to an extent, but I do think that you know, for me, like the influential advancements are maybe a little bit more around what we've learned to do and not to do for the next pandemic. And in some ways, I know that might sound a little bit generic, but I feel like the not to do list has is pretty clear that I feel like next time it will hopefully save more lives. And I think I'll just keep it simple at that.
Yeah. And I'll add that I think some of the public health things that that maybe we knew already, but we have, as a community, come to appreciate how powerful simple public health interventions can be. At least for me, has been very humbling. And the whole concept of asymptomatic or pre-symptomatic people and spread of infection, I think, has been a discovery that has affected some of the approaches we're taking to try to prevent infections. All right, I'm curious to hear, other than your own.
In my hospital, I mean, how important public health measures, that's probably another big thing that has been, I mean, when this thing started, and I know my friends and my family, and they look at you, you're going to the hospital, you're at very high risk of transmission of this thing, and you feel like a pestilent person. And in some way, and you go back home, you undress and you redress, you start working with scrubs that you have not done since your residency time. But when the hospital started the universal masking policy, and I think the cases among healthcare workers plummet. And now in Philly, in my hospital, the seroprevalence among healthcare workers is 2% of positive. In the city of Philadelphia, 5%. It's safer to work in the hospital than to work in the city of Philadelphia and go to the supermarket. So masking and washing your hands work and prevent infection. That's another big lesson from this epidemic. Public policy works. I agree with you completely.
So I want to hear about other drug candidates and vaccine candidates besides your own. Which ones are you excited to hear about or see the results from trials or studies that are ongoing?
Yeah, you know, I think for me personally, I just think that as we've you know learned a lot more about the disease pathogenesis and specific targets and you know some pathways that are activated during the disease. I am agnostic to any particular therapy, but I think as all these different things progress through, honestly, like I just it's like learning new things. There's so many people focused on many different, whether it's the IL-6, it's the JAK inhibitors, IL-7, neutralizing antibodies. There are so many targets out there that hopefully as some of them start reading out soon, it would really be interesting to see where we land. And I think as we learn more things, then we recognize, oh wow, we didn't even know these other aspects before, or it'll be interesting to see where it goes. I think especially in that critically ill population of patients that have more severe disease, what else can we do to actually improve mortality in that group and disease severity as well?
In the vaccine field, I think there are many different technologies that are being tested. I don't think there is going to be a clear winner. I think, and in fact, I think for humankind, we shouldn't put all of our eggs in one basket. And I think some vaccines might work better for some populations than others. Maybe the elderly needs some type of vaccine, the young needs other type of vaccine. And this is not going to be a race that there is one winner. I hope there is several winners, and will be to learn what are the particular populations, what do you need to vaccinate a particular group of individuals to have a more immunogenic vaccine. And I am excited about protein-based vaccines. I am excited about the mRNA technology. Moderna and BioNTech there is a new way of developing vaccines that are very interesting. The Chinese published today a paper about adenovirus 5 that have the clone they have the spike protein with a vector, with an adenovirus vector. It was published in the Lancet this morning. So there are many different technologies, and we need, I hope several of them make it. And then we can optimize how do we vaccinate people, which vaccine is better for children, which vaccine is better for women, which vaccine should be used in the elderly or immunosuppressed individual. Because I don't think one size fits all. And that's what I think I'm more excited about it, trying to learn how to use these different products.
What do you think the probability is that we'll have a vaccine available for use in 1 year?
I hope so. The country is putting a lot of resources. The NIH is putting a lot of money to developing these networks. And if what has happened during these first 3 months of this pandemic is a signal of what is to come, I think we as a human race, we will step up to the challenge, and there will be a vaccine. Hopefully, we will prove that it's effective. Then those engineers have to prove to me that they can do 6 billion of those vaccines, so we can vaccinate a lot of people. And that we can do, we as healthcare workers can deliver that intervention to a lot of people. One of the other things that we have learned in this pandemic is that our health systems are not good enough, and that the disparities between the people that, like me, or that have, and they have good access to medical care, and the people that do not have access to good medical care, they get accentuated in a pandemic situation. And you see that people that have diabetes and obesity, hypertension, cardiovascular disease, that are more frequent among people that have poor access to care, and with a lot of food insecurity, they come into full view for everybody when in a pandemic situation. That's very hard to see, and we need to have better health system for everybody.
Sarah, I think we should wrap up, given the time. I'd like to say again, we have been very fortunate. This has been a wonderful discussion, and it's clear we need to have subsequent discussions as we learn more about the failures or successes of these treatments. Sarah?
Thank you both. I've learned a huge amount, and it's really inspiring to hear about your work and to hear your thoughts on where we're headed. And I continue to look forward to seeing the developments coming from the both of you. Thank you.
Thank you very much for the invitation.
Thank you. Have a great weekend.
That concludes.
Congratulations, Anu, in that paper.
Yes, Anu. Congratulations.
Very nice.
Thank you. We're going to end the discussion now.